Remember, the content on our show is for information only.
It is not intended to provide medical or other professional advice.
Hello, and welcome to Placebo, the podcast where each week we review interesting news articles about healthcare or medicine. I'm Logan Richards, and this is my sister.
And I'm Kaylin Richards, and I was thinking, do we just review them? We review and discuss.
Okay, we review and discuss interesting news articles about healthcare or medicine.
Yes.
And this week's article will be...
Undead, in quotes, genes come alive days after life ends.
Okay. And the source of this article is something called Science Mag. Now, convince me that that's a credible source.
It's very credible. It's the magazine associated with the what does AAAS stand for? Hang on, let me find it.
I'm not convinced yet.
Oh, AAAS. Well, they don't say what it stands for because it's so obvious, apparently.
It's the American Association for the Advancement of Science.
Oh, okay, there you go.
Now, why is that a credible organization?
Here's some facts about Science Magazine and its publisher, AAAS.
Okay, good.
So they're talking about how science is important first, and then they're talking about Science Magazine is important because it publishes the very best in research across the sciences.
Now, do they publish research as well as these news type of articles? Looks like they also have journals.
I think they do both. They have journals in specific things. So they're talking about some things, some articles that they've published. They published a story about the entire human genome.
Yeah, I think they have a scientific journal just called Science, and I think that's
more general. They published images of the Martian surface. They published the first studies tying AIDS to human immunodeficiency virus. Those must have been a little older. That's well known at this point.
All right. I'm trusting it a little more. It still has too many ads for my liking on their website.
Yeah, well, otherwise it would cost money. It's a nonprofit, it says.
Okay. Well, they got to make money somehow, I suppose. The other thing about this website that made me question it originally is that the way that it's set up is like one of those kind of ad revenue sites where they just have random articles, and then at the bottom where it says more from news, and it has like these weird looking pictures with clickbait articles at the bottom.
But it's a real website.
Okay.
It's true.
Well, regardless, they reference a study which we think is
And also, I read the actual study as well.
Right.
So, okay. Undead genes come alive days after life ends, and it's got a real sad picture on the top of a dead rat with a fly on it.
Yes.
Which makes me sad. So
it doesn't appear to be one from the study, though.
No, because the study did fish and mice. So they found a stock photo of a dead rat.
That could be a mouse.
The caption says rat. All right, this study is written by Mitch Leslie, who is a writer for the Science Magazine about cell biology and immunology. He's not a researcher himself.
Okay.
So you enjoyed, I think, the start of this article a little bit. You commented to me on it.
because the writer puts Plato and Blue Oyster Cult into the same category of great thinkers.
Well, music is a great way of thinking as well as philosophy.
I think it would be a very broad category.
Yeah.
But I could see how they could both be great thinkers in their own right.
In their own areas of thought.
Yes.
They talk about how, people have thought a lot about death and what that means over human history.
Sure.
From Plato to the Blue Oyster Cult and beyond.
No one has thought of it since Blue Oyster Cult.
They were the last people.
They were the last ones.
But so this study that has come up is looking at how one aspect, at least, of life continues after animals die, and presumably after people die, I guess, too.
Sure.
And it's that genes stay turned on and productive for up to, I think, 4 days in the case of the fish that they studied. And they studied the mice for 2 days, 48 hours, and they were still producing at that point, but tapering off.
Okay, here's my question to start, because this is all about gene expression.
Yes.
And I guess I don't really know what that means, at a practical level.
Okay.
So what's an example of a gene? It doesn't even need to be one that is dead, but what's an example of gene expression that I would be able to connect to?
Okay. What a gene does when it's active is it makes a protein.
Okay.
Okay, so it
instructs the cell to make a protein?
It instructs the cell to make a protein or some type of product like that.
Because genes are in the nucleus of a cell, right?
Right.
So that means that if they're being expressed, then it's almost like
that
set
segment
of
the DNA
instructions or something?
Yeah, it's like a recipe. That segment of the recipe for everything that you'd ever need to do your whole life and after is active, and something is happening. So generally something is being produced, and they can measure the way that the RNA is producing or is structured, and they can measure if it's active or not.
Because there's all sorts of genes in there, and only the ones that are being expressed are the ones that cell is using. Is that right?
At the time.
Okay.
Yeah. Because the study goes into, there are some genes that become active when you are hypothermic to help your body warm back up, for example.
But they're not active.
But like right now,
in any other case.
I'm not hypothermic. I'm a little
close.
cold. It's a little cold here.
She's close to hypothermia.
I have my coat and my scarf on.
But they haven't activated yet.
But I'm not, yeah.
Okay.
So as an example.
So that's interesting. So what they're saying is that there's still some cellular activity, basically, 4 days after death.
Yes, which is interesting to me because my picture, and I guess reading the actual research article, I think I was wrong, but my picture is you die, and it's a moment, and once you're dead, right?
Well, there's a reason Plato was brought up, because this is a very philosophical topic.
Right, okay.
What does death actually mean?
Well, yeah, a lot of people have thought of these things as they said. But in the research article, they basically said, okay, these are all the things that we found, and we'll go into this in a moment here, and we really expected to find all these things were still active after death. They did find one thing which they thought was particularly surprising after death.
Okay.
And that it was still active, but
yeah.
Okay.
So the study was done at the University of Washington in Seattle.
Ooh, Seattle! Although I found that I think the maybe statistical analysis was done at the University of Washington in Seattle, but at least as far as the mice data that they got, the mice were in Germany, I believe.
Okay.
So
well, good. Collaboration is what we need.
International collaboration. So anyways, yes, but the lead researcher was Peter Noble, who's from the University of Washington.
Okay. Says he's a microbiologist.
I'm assuming that's correct then. So they're testing a new method to calibrate gene activity measurements, and they decided to focus on this post-mortem measurements.
I never felt like it was explained why. Was that just easier for them? I get that they want some gene expression measurement or whatever.
Okay.
So why did they choose dead things?
So I think he said it's an experiment of curiosity, which is a little bit creepy considering, we killed mice and fish to just see what happens. That's not great, but
I guess he's figuring there's plenty of experiments where they already end up that way.
I know, but it doesn't seem like you should do it just because...
It doesn't sound like he did it.
He didn't. Some people in Germany or something did. So here are some ways that they think it might be useful, though.
What might be useful?
Oh, the data that they got from this. Why you were asking why they did this study.
Well, I'm just asking why they did it on dead things.
Oh, this is why.
Okay.
Because, for example, preserving organs that are donated, it would be helpful to know what genes are active in the organs, taken from a person who is dead.
Okay.
And how that would affect the person that the organ is put into.
Sure.
And then also they talked about, as far as for murder victims, for example, it's really important to pinpoint accurately when they died to solve a case. And it can be helpful if they can get a really specific time of death. That could make or break a case, right?
Okay.
I think that I've watched a lot and read a lot of mystery programs.
You're watching too many crime dramas.
And listen to Serial, a lot of stuff, right? So, and I think one thing that comes up in a lot of these situations...
Would they have been able to solve Serial if they knew the exact time of death?
Well, I don't know. But a lot of times they'll have the coroner or the pathologist, and they'll say, okay, time of death was from midnight to 4 a.m. And that's a huge window. And if you can... If 4 of your suspects have alibis for half of that time, but not the rest, you could, rule them out if you knew it was really only from midnight to 12:30.
Sure.
Anyways, those are two examples of how this data could be useful. Although
But didn't The people at UW, right? Peter
Yeah.
Noble, they didn't do that originally to figure out those two things.
He said it's an experiment of curiosity to see what happens when you die. That was the quote from him.
But those two examples that you just gave as something that why this would be useful, I think those were given by someone else.
That was discussed in the article. They weren't examples given by someone else, but they were discussed in the research as, okay, now that we have this data, this is what we think it could be useful for.
Okay.
I think until they did the study, they didn't know what the results would be, so I guess they didn't know how it could be useful.
Sure.
Which is part of science. Again, I just don't like the idea that they're killing the mice.
Well, at least they're making use of it.
Now they are, but what if it wasn't useful?
Well, I think the mice were probably already dead from some other experiment.
No, they killed them for this experiment.
Oh, well.
So, they talk about how scientists, when they were looking at tissue from human cadavers, they'd noticed that there were post-mortem activity of a few genes. So they knew this was something that happened.
Okay.
In this study by Dr. Noble, they looked at over 1,000 genes in these mice and zebrafish to see what was active. The human studies had really been, it sounded like more incidental when they were doing other studies, that they just found that these genes were active.
Okay.
So, as I said before, they looked at the changes for 4 days with the zebrafish and 2 days in the mice. They assumed, I think as you and I would, that the genes would pretty much shut down pretty quickly after you die. Like they thought there would be some expression after death, but it would ramp down pretty quickly.
Right.
Right. What did they find, brother?
Some genes remained active for 4 days after death. You already said that. But...
Yeah, I did. So, and what was even more interesting is that instead of just like tapering down slowly over 4 days, some genes were dormant or inactive, and then after 24 hours, say, then they kicked in.
And so did they talk about like why this happened or what's important?
So,
does this mean they didn't actually die?
No, they did. But here's what's interesting. So a lot of them are things that you would expect. The type of genes, like I mentioned, a gene for hypothermia, they saw that. Also genes that would be active when you are hypoxic, like your oxygen levels have gone down.
So it's like the body's trying to save itself kind of thing? Like all those genes?
So here's what's interesting. The body didn't know you died. The body's just like, oh, you're not breathing very well. Oh, you're too cold. Let's try and get you warmed back up.
So what's the official like death definition then here? If the body doesn't know,
well,
is it?
I don't know. What do you?
The nervous system turns off.
The heart has stopped beating. I don't know. Consciousness is gone, but it's a mouse. How conscious? I don't know.
Well, okay. So interesting. Right?
Yes.
So the body doesn't actually.
So these cells,
specific
individual cells, don't necessarily know that you are dead.
Right.
For a while until they don't get, nourishment over too long of a period of time.
Which is pretty crazy, right? It kind of makes it sound like an ant colony where like everyone's doing their own thing and have no idea what anyone else is doing, but somehow it all works together perfectly. The
body knows like, whoa, something's wrong. You're way too cold. We got to work on getting you a warmer.
But that sounds like the nervous system, right? Because doesn't that... in order to sense that you're cold, the nervous system has to know that you're cold, and then it has to go up to the brain to tell other things that you're cold, right?
And they were specifically looking at brain. In the case of the mice that they did the study with here, they looked at their brain tissue and also their liver tissue. So they were looking at specifically the nervous system in part.
So it must have just been the heart had stopped or something.
So, they found that like the emergency type genes were what they saw initially. A lot of the things that help with inflammation, with the immune system, with counteracting stress in general. Okay.
Well, it is really interesting, though, even just regardless that they didn't actually know what was going on.
Right, until like, the cell hasn't received any sugar for this amount of time or whatever.
Right.
The most surprising thing that they found, though, was that the developmental genes, is what they've talked about, were turned on after death. So these are genes that you don't normally use throughout most of your life.
Okay.
They are used when you're an embryo and just starting to be formed. And then they're not active after you're born. But they become active again after you die.
And they don't know why? Or do they know why?
Well, so let's see what it says. It says they aren't needed after birth. One possible explanation is that the conditions in the newly dead corpse resemble that of an embryo. I'm not quite sure what they mean by that. What are your thoughts?
I have no idea.
In that they are not... No, that doesn't make sense to me. I was going to say they're not breathing. An embryo, though, is getting blood through the placenta, and oxygen in that.
I think you have to know more about death and embryos than we do in order to make that connection.
They don't explain that here. I'm looking back at the actual study to see if it's explained in a way that doesn't use 1 million words. Like an actual word here in the study is glycosylphosphatidylinositol anchor 2.
Of course.
They say the same thing as the article. A possible reason for this activation is that post-mortem physiological conditions resemble those during development. So
you have to be a microbiologist to get that.
Maybe.
To get that metaphor to work for you.
So one thing they wanted to point out here, though, is that people that receive organ transplants have higher risk of cancer. And they thought that the reason could be because the genes that are associated with the embryo type genes can also...
These developmental ones
can also promote cancer, because I think they're promoting more rapid growth or things like that.
That's interesting.
And so perhaps the reason that people with
so
transplanted organs,
transplant,
it's had this burst of these embryonic genes which could cause cancers.
I assume once that organ is back into its normal conditions and being used by someone else, then those developmental genes should
go down. I would assume so, but I guess they don't know that. They haven't studied it.
Right.
Because the reason that the genes are activated is a little bit unknown, right? Because the stress ones, it makes sense, right? The body is reacting to the stressors that it's experiencing.
Right.
But it's not really clear why it would be turning on those embryonic genes.
Maybe they're hoping to grow some, I don't know.
Maybe when you're an embryo, it's just before you're alive, and when you're dead, it's just after you're alive.
Okay, so this is, in the end, did they actually develop a method for telling which genes are expressed and when they've been started to express?
Yeah.
So is it reasonable that this will be used for these kind of criminal cases too? Because I didn't read a lot about. They obviously know when a gene is being expressed, but how are they determining at what time that gene expression started in order to pinpoint things like death?
So I think obviously they would have to do more studies in humans, right?
Sure.
Because you would need to get how things are typically expressed in humans that have died.
Yeah.
Which, I mean, even just looking at their two, the mouse and the fish, they had different kind of trajectories of how their genes were expressed from the different species. So I would assume people are different as well.
Yes.
So my thought is you would need to do enough studies to get the average trajectories of different genes in people. And then if you had, a body that was discovered, you would take a tissue sample and then test it out and see exactly where it was at that time, and then you could work back from that.
It doesn't sound like they're carbon-14 dating or anything, though, like where there's a certain amount of something and they know how it degrades over time, so then they can figure out.
No, because it's the opposite, because like some things, 6 hours after death, this one starts, and then it
so maybe they're measuring
maybe
they're measuring like amounts of proteins
after
a certain amount of time.
Yeah, exactly.
Okay.
So it is kind of carbon-14.
But the opposite direction.
But it might go in the opposite direction, yeah. Depending on
That's our understanding.
On when you find the person. So one thing to point out is that the paper this was based on is not completed its peer review yet.
Oh, how did it get published?
So it's on
Is
this.
It on one of those open source ones?
It's called a preprint server.
Oh.
And I'm not sure how you're supposed to pronounce it. BioRxiv.
Bioprescription IV.
Maybe. I'm not sure how to pronounce that. Prescription IV.
Biorexiv.
So it's still undergoing peer review. And this article was published in June.
Okay.
The study was published put up on the preprint server before that, and it's still, when I looked at it now, it still hasn't finished its peer review. So peer review takes time. So one other thing about transplants that I wanted to bring up, it mentions how you could tell potentially like how successfully a transplant is going to be if you tested the organ going into the new person, and saw, it's had, a lot of increase of this embryonic
so it
DNA.
might be less successful.
It might be less successful. It might be not as good a one to do versus one that was very fresh, maybe would have less of that turned on.
Well, that's not surprising because I think it's already known that one that has is less time since death works better, which is why they rush so quickly to do organ transplants.
Right. Sometimes, the quicker sometimes even though they rush, you still can't be as quick as you would want, I guess.
Right, sure.
Yeah. So let's see if we can wrap this up, huh?
Yes. I don't know how to wrap it up.
Okay, I'm just reading the end of the article here. And another final point that they make, and I think maybe this might be the key to the embryonic DNA or why it's being turned on, the genes. Mm-hmm. It says that some genes are inhibitory on others.
Sure. In
life. And maybe once the organism has died, they become inactive.
And there's no inhibitory genes.
Right. And so maybe that's could be a reason why the embryonic genes turn on, because there's nothing stopping them from being there.
Oh,
but then why would the other one turn? It doesn't make sense.
So this is quite new information.
Yeah.
With some interesting applications, possible applications.
The ending is a quote from the Dr. Noble, the lead researcher. He says, "The headline of this study is that we can probably get a lot of information about life by studying death."
Sounds like a philosopher.
Yes, I really hate the idea of killing the mice just to study about their death.
Well, perhaps they can use mice that have already died in other studies, and then they can continue to be useful in the advance of science.
Yeah, I think the reason that
they make you feel better.
I think that's impossible because they wanted to have controlled subjects that had similar genetics.
Perhaps as the research advances, though, then they can start to make use of animals that have already died.
Okay.
Do you feel better now?
No. So, anyways, I think this is a really interesting article and really shows kind of how blurry the line is between life and death, and more blurry than I think I was thinking it was.
It certainly seems that way, yeah.
Yeah.
Very interesting.
All right. Well, if you enjoyed this or were interested in this article, we'll have the link to it in our show notes.
Yep. We have a Facebook page about Placebo Podcast if you're interested in contacting us or giving any feedback.
Our New Year's resolution is to be consistent in posting our articles on that as well.
Oh, okay. Perfect.
This is our podcast resolution.
Also, more information for you. We've decided to change the release date to Sundays since we weren't very good at Wednesdays.
It's really hard in the middle of the week. So, Sundays from now on, you can look forward to seeing a new episode and start your work week off right. Thank you to James Aiken for our music.
And thank you to all of you for listening. We'll see you next week with a new episode on Sunday.
Nice.